So now let's talk about drug discovery and designing of drug. Now you might think what is the process of designing is needed here in the case of drug discovery. Of course, a drug discovery involves finding out a drug, but designing a drug means to optimize the properties of a particular drug. That is, sometimes it might have a binding property and a binding energy of one particular value. But if you slightly change the molecule or if you modify the drug probably, its binding energy can be reduced later, even little more down. So that process of discovery and designing with the help of computers, that's what we're going to do today. Firstly, let me ask you, what is a drug? Drug is any chemical compound which is being used within a body. It can be used for different purposes. Now, either for the diagnosis, treatment, or the prevention of a disease, or in any abnormal condition. To overcome that abnormal condition, we might use a drug. Or thirdly, you can use it recreationally for the effects it gives on the central nervous system, such as in the case of narcotics. And you have heard the term drug abuse. That is, most predominantly various drugs or various chemical compounds are being taken as recreational factor, or you take it recreationally as a to get some hallucinations and to be outside the world for some time. Those are those which come among the category of narcotics, or which we say the frequently used drug abuse. But the term drug doesn't always come to the same aspect of drug abuse exact, like, because there are certain, even the common paracetamol that we have against fever, that is also a drug because it is helping us to overcome a particular situation. So, any chemical compound which serves these three purposes come under the category of drugs. It can be either for the diagnosis, treatment, or the prevention of a disease, or to overcome an abnormal condition. For example, if you're getting poisoned by a snake, you might give a anti-venom, that is also a drug. Or you might also take a drug recreationally, as we had mentioned earlier. Now, what is drug design or the rational design? Drug designing is an inventive process which involves the finding of new medications based on the knowledge of a biological target. You know what is a biological target? A biological target is some component of the disease causing agent which we can block or which can be inhibited with the help of the drug. Basically, it involves the design of small molecules which are complementary in shape and charged to the biological target with which they interact and therefore will bind to it. So, in the process of drug designing, what you do, you get a biological target and you will design or produce. You know what's designing? You know what's designing in the case of cloth fashion industry now. So the same way here, in the if you have a particular drug, you design it or modify it in such a way that it becomes complementary in shape and charge to the biological target. That is, it will bind to the active site or the relevant portion of the biological target so that it will prevent the biological target from doing its particular function. And when you use computers for the drug designing, or the that is referred to as a computer aided drug designing, or simply called as CAD. Now, what is the biological target? Biological target plays an important role to destruct the disease which has been caused by this pathological agent. Now, you might have, what is this? It seems to be very much complicated when I define it like uh, it's it is something which will disrupt the target for mechanistic drug and the process and all that. Let me just show you a better, easier slide so it's easy for you. Now, all the biological targets that we consider here, it could be either enzymes, it could be receptors, ion channels or transporters, or even nucleic acids. Now, sometimes consider a disease is being caused by a by a microbial agent, and that microbial agent has an enzyme which will help it to propagate within the host, or it has an enzyme which will cause a drastic or abnormal condition within the host. So if we block that particular enzyme, okay, in either of the cases, if you block that particular enzyme of interest, what happens? You can control the particular disease. So that in such a case, what happens? The enzyme becomes a biological target. Now, in the case of HIV, we have HIV protease, okay, that can be inhibited so that the replication of HIV will be inhibited. Okay, so different microbes have different enzymes which, if we inhibit them, their replication can be prevented or the disease progression can be prevented. So enzymes serve as biological targets. And another thing that we have is receptors. The receptors of the cellular function can be blocked, as in the case of rhinoviruses. That is, the receptors are molecules on the surface of the cell with which the pathogen can enter into the cell. Or they identify the cells where they need to attack using the receptors. So by blocking the receptors, what happens? You can prevent the effect of that particular pathogen. Or sometimes they might be involved in various signaling pathways. So breaking the ion channels or transponders or the transporters could also help to interrupt the signaling pathway and also the progression of the particular disease. And in some cases, you can also see that nucleic acids can block the synthesis of undesirable proteins at the nucleic acid level. Example, even in the uh, tumor as well as in antimicrobial activity. Then, so what is CAD? CAD is a computational chemistry. It is defined as CAD or the computer aided drug designing, and it is a chemistry whose major goals are to create efficient and mathematical approximations and computer programs that will calculate the properties of the future drug molecules and thus help in the process of drug designing and discovery. So, simply speaking, when you use computers to make some mathematical calculations as well as computer programs, so that we can find out which is the prospective or which is the future drug molecule and which will help in the process of drug designing and discovery. So, the use of computers for drug discovery as well as drug designing is referred to as CAD. Now, why do we have to do CAD? The process of drug discovery and development is usually found to be very long and which is being found to be very difficult one. And the costs are also being found to be increasing. Now, approximately we can say that about 10 years and about $100 million are being needed to take a new drug to the market. And of course, there's a lots of competition between the different pharmaceutical companies and they need to save time as well as money. And so in such a case, if a computer comes and helps them to find out the drug fast or the prospective drug fast, that would be helpful. Now, previously, if we do not have CAD, of course, this could be been found to be even little bit higher. That is, instead of 10 years, it will again further progress. Now, in the chaos of COVID-19, of course, we are not going to expect it for 10 years. We do expect it like within a period of 2 years or something, like we will get a drug. That's what, a vaccine, of course. That's what we are expecting. So let me just take you to what is a drug discovery pipeline usually. Usually, when a new disease comes, you need to find out what is the target. What is a biological target? Is it a protein or it is a lipid or is it a ion channel or something which has to be disrupted, or is it a receptor that has to be that has to be blocked? That is the first step of drug discovery. So the target of the biological pathogen is being identified. After that, you will create hits, or you will take different drug molecules and find out which all will interact with the biological target. Okay, and after that you find out the lead molecules. What are the lead molecules? The molecules which will interact with the biological target at the most beneficial way and with the minimum of the binding energy. This mainly constitutes the early stage of research and discovery. So the process of this, okay, this drug discovery and all that will be aided with the help of, if it is with CADD, the amount of years that we need can be reduced a lot for a drug discovery. After the lead molecules are being found out, what happens? You need to do clinical trials. First, preclinical studies have to be done in animal models. After that, clinical trials will be done. The first phase will include about 20 to 80 healthy individuals. The second stage of about 100 to 300 patients. Then it becomes again 1,000 to 3,000 patients. So the time duration for these clinical trials, each phase consists of about 1 to 2 years. So here 1 to 2 years, here 1 to 2 years, and here 2 to 3 years. So the approximately speaking, we have almost 2 plus 2, 4, 4 plus 3, 7 years which are being needed for the clinical developmental phase itself. And then again we have 1 to 2 years for the approval phase, and only then the chemical synthesis of this is being of a drug is being usually done. This is a normal drug discovery profile. Now you might think, what is the difference with the vaccine in this? Of course, a vaccine also has to undergo so many years of clinical phases and clinical trials and all that. But when it becomes an emergency, we are trying to reduce. Now, we got COVID-19 in the case of November 2019, and now we are in a stage of about the first phase of clinical trials of these two things are bypassed. That is, preclinical studies in early stage has already been completed within before a period of 1 year, and we are going at a faster rate. And you can see at each stage the cost of, or the millions of dollars which are being needed, is also being found to be very. And if you go to look at the drug discovery and development timeline, you can see that if you have 10,000 compounds which are being used for drug discovery, 250 of these go into the preclinical studies. And then 5 compounds go for clinical trials. And then the FDA review is being carried out, and one FDA approved drug is being involved. So you can look the amount of years which is being taken for all the entire process of drug discovery. So earlier in olden days, what is it? We used to take each and every plant, okay, or you will take an extract of a particular plant and give it for the patient, and you see, okay, fine, is it working? If it's working fine, you take then further, then you take that particular extract, and then you try to isolate which component of the plant was responsible for that. Then you chemically synthesize it. It takes even more time. So, but when you go with computer-aided drug discovery, you can identify exactly, okay, in a faster rate, which are the molecules which will bind with least binding energy with the biological target. Hope so you got an understanding of the drug discovery, or the relevance of CADD in drug discovery by now. Thank you for this session. Let's see in the coming sessions the processes of molecular talking.